So we've got 13 papers open. This is what I was able to pull out of the BioRxiv this week. Let's have a look at what they say. All of these are preprints this week, which means things that have been probably submitted to some journal and may be in the review process, but they're going to change a little bit on the way between that process and full publication. So you got to take them with a little grain of salt, but it's a little look at what is coming down the way. Okay, with that aside, let's go into the first paper on the first tab. So this one I think is absolutely fascinating. So it's an older study, 2013 to 14, but they're looking at the transmission of bat coronaviruses. Now, not the same ones that cause SARS or MERS, but some interesting relatives that we didn't know could infect other kinds of animals, and it turns out they can. So they're looking at these coronaviruses, and they're looking at how they spread along the human supply chain, basically the food supply chain. That's what we're looking for here. And so if we go on here, the pictures in this one are wonderful, which is part of why I pulled it up. So among other things, this tells us these are field rats, and she's pulling the skin off one, so she's basically shucking rats instead of corn cobs. And here's a cage with them at one of the live wet markets, and this is all in southern Vietnam. Here are some Malayan porcupines, as in show, they're in their little cages at one of the porcupine farms. And here's a bat guano farm. So they put up these little palm leaves in the top, and the bats will then roost in amongst these, and that's the guano down there on the floor. And so they're sampling these sites. They're sampling basically from the farm to the table. Yeah, how trendy, right? And this is the key right here. So they're showing the prevalence of these coronaviruses. This one shows which ones they found, and they're going to be some of these beta coronaviruses and some of these alpha coronaviruses. The alphas are related to other bat coronaviruses. They're not particularly close to the human alpha coronaviruses that we know of. The beta coronaviruses kind of are all around and in between the pathogenic beta coronaviruses, but not exactly those. But what they show is that you start out with very low levels of infection at the first trader, who's basically taking them straight from the farm. The amount of infection increases as you get to the large markets, which are collecting up animals from the little traders. And by the time you get to the restaurants, kind of the final step before it's served, you're going from 20% up to 55% infection in these animals, and reasonable numbers that were sampled in each one. And the it's also moving around from place to place. We're getting, as the animals are transferred, the virus is getting transferred, and you're getting clusters of related virus. So here's 1, 2, and there's a third big cluster over there. And these things are jumping from one species to another. So you've got them in the bats from the guano farms, but then you've also got the same coronavirus jumping with minimal mutation to Hystrix, which is the porcupine, and to Rhizomes, which is one of these rats, and then to Rattus and Bandicota, which are the two kinds of field rats they looked at in the study. So this is a microcosm of exactly what we think happened in the SARS coronavirus and why wet markets are a particular risk. This one's in Vietnam. This one's from a couple of years ago, but if we had the same data from the wet market in Wuhan and all the suppliers leading up to that, chances are we would be able to reconstruct exactly what happened. All right, on to the next one. So this is a neat paper. I'm not exactly sure what it shows. So they're purporting to show actual SARS coronavirus on the outside of a cell. And boy, do they look cool, and they're sure right about the correct size. And they're actually showing the cell kind of folding over and wrapping up a big parcel of viruses to potentially bring them in. Here's one of those little folds, and you can see what look like maybe viruses on the edge of it. So there isn't a secondary stain or confirmation in here to actually show that these things are definitely little viruses, but they look virusy, and they're pretty neat images. So I just thought you might want to see that. Here's the actual paper up here at the top. There we go. All right, next one. So this one is looking at infection in the brain. There are actually, I think, 3 papers this week that are looking at infection in the brain. And this one is saying that, well, if you look, most of the brain, doesn't really seem to have the right receptors. But the human hypothalamus and connected regions, right there at the center, have very nice high levels of ACE2 and TMPRSS2. So these are potentially sites that could become infected. I don't believe there's any direct evidence in this paper or any of the other papers of the hypothalamus becoming infected, but it's a thing you wouldn't necessarily know to look for otherwise. All right, next one. So people have had this question. You have antibodies in the bloodstream, and you have neutralizing antibodies. The neutralizing antibody test is hard to do and kind of slow and has to be done in a specialized lab. The regular how many antibodies do I have test is basically like a pregnancy test. It's pretty easy, just needs a drop of blood. And so people want to know what's the correlation between these. Well, this intrepid group actually set out to find out. And so they're correlating the amount of total antibody against the amount of neutralizing antibody. And they're looking at antibodies, I think, to the whole protein and to just the receptor binding domains. And they're showing a nice sort of normal response there. And the eventual conclusion here, the authors and title of the paper, if you want to go look that one up. But the eventual conclusion is that they found, there we go, when the probability of a reasonable virus neutralizing titer, amount of antibody, greater than 160 or equal to 160. Now, we'd actually like titers higher than this. With convalescent patients who have had moderate to severe SARS, the average is a little higher, maybe more like 500 in most of those. Seems to be like the baseline. But you got about an 80% chance of having that if you have a regular antibody titer of greater than or equal to 1 in 1,350. So neutralizing antibodies are, in other words, something like 10% of total binding antibodies is roughly what they're showing here. And that's useful. This is a useful piece of information. It's not 100%, it's not a direct correlation, but it will help the quantitative tests like the ELISA to be used to try to at least estimate the parameters on the neutralization, but we still need better neutralizing antibody tests that can be performed outside those specialized labs. So that's the thing that's going to be coming. Now, the next paper is showing here, so it's the effect of some of these medications. So people have been asking right from the beginning, what happens if I'm taking ACE blockers? Because angiotensin converting enzyme 2, that's the receptor for SARS coronavirus. And there's also an angiotensin converting enzyme. You could call it number 1, but it's just it's just the original. It doesn't have to have a letter or a number after it. People take inhibitors of this because ACE1 is going to raise blood pressure, ACE2 is going to decrease blood pressure. They're both parts of the same system, and substrate gets passed from one to the other, and so they're just sort of pushing and pulling, basically, regulators of this process. And there are a lot of people on regular angiotensin converting enzyme blockers. These are 2 of them, that thing, and losartan, by the look of it. These are both drugs that block ACE1, and so they're looking at the effect on ACE2, and as you can see from the bars here, there is no effect anywhere on ACE2. So people were worried that knocking down ACE1 might lead to more or less ACE2, and it turns out that the answer is neither. It just it has no relevance whatsoever. However, they also put in, I think it was an siRNA, so a way of knocking down the amount of actual angiotensin. So this is the thing that ACE and ACE2 are converting. And when you knock down angiotensin, you actually end up with lower levels, if I remember correctly, of TMPRSS2 in the lung. And so there's our little starred thing over there. It's not extremely low, but it's slightly lower. And so I don't know. I think the conclusions on this one may get toned a little bit back in review, but the data itself seems like it's okay. So we'll see what happens when that comes through. But that's interesting. Okay, first of 2 papers with a very important finding. So they're showing in both of these papers that a protein called Neuropillin is they're trying to avoid saying it works as a functional receptor for SARS, like equivalent to ACE2, but it seems to work when you knock ACE2 down. It still seems to lead to more entry, and it enhances entry even when ACE2 is there, if I understand the papers correctly. So it's pretty much a second receptor for SARS coronavirus 2 that operates in the brain. Interesting. So shown here up at the top, this is the neuroepithelium. This is where those smell receptors are going to be, and this is hooked up to the nerves. They're going to go into the olfactory bulb of the brain, which is that little knob right at the front, which is where you smell. And the brown stuff in here, or reddish brown stuff, this is going to be staining. So they've got an antibody, and they've done a in situ hybridization by the look of it. And they also show that so you got very heavy infection of that neuroepithelium. This is something that we speculated could happen from a previous paper that showed that there were high amounts of ACE2 and TMPRSS2 in that area. And this shows that, yep, it sure does happen. And this is the area kind of at the back of your nose that they would be able to sample with one of those swabs that's pushed right back in there. So those swabs, they're actually doing what is more like an influenza test. But in this case, it does probably pick up at least some of the SARS coronavirus too. And they show that this is infected, and some of the neurons inside do have a little bit of that staining. And so the infection is spreading from the neuroepithelium, which is probably going to not work as well, hence the lack of smell in some people, to the actual neurons of the olfactory bulb. And so infected neurons, infected brain, potentially very serious complication. This might explain some of what is seen in SARS coronavirus 2. So interesting. Here's the second paper, and these two are coming out at the same time in the preprint server. They may be coming out in the same journal. I don't know if they're doing something like that, or if they're just going to try to be racing each other. But this one is also showing that Neuropillin 1 is important, and this one goes a little more into the mechanism. So Neuropillin 1 is good at binding proteins that end in RRAR, and after the spike protein is cleaved by the enzyme furin, it will have exactly that end, that C-terminal end of the S1 region. And so that is the part that Neuropillin can actually bind. And so it's a potential use for that polybasic site. We know it doesn't lead to an extreme amount of additional binding, but it does increase the amount of binding at least a little bit. Entry can still work without this polybasic site, and they do mutate this site when they're trying to solve the structure of the protein. It just keeps the protein from falling apart, basically. So that's specific to SARS coronavirus 2. It's also common to many, many other coronavirus S1, S2 junctions. So this is one of those things that, pardon, people have said is evidence of genetic engineering, but it's something that's actually pretty common in coronaviruses in general, and so that's where it is, and that's what it does. The next one over here is looking at a very interesting effect. This is the SARS coronavirus, and it's got two proteases, the papain-like protease up here and the main protease, or 3C-like protease, down here. Papain-like protease cleaves this little protein called interferon response factor number 3. And that is one of the major ways that a cell communicates that it is infected. So there are various detectors in the cytoplasm. They will pick up the traces of the virus, different aspects of the virus, and then IRF3 is one of the main molecules that is going to carry that signal into the nucleus and lead to changes in transcription. And so the virus is going out and actively destroying possibly the most effective messenger that would lead to sort of exposure of the virus. So that's very interesting. I had not seen anything like that before. So that's the papain-like protease that's actually cutting in IRF3. And the main protease is cutting out two other proteins. It's snipping them, this TAB1 and this NLRP12. They would both be involved, but to a lesser extent, or maybe just less directly. But these look like potentially evolved traits of the virus to shutting down the intracellular immune response. And so that is very fascinating. Here is the actual paper up here. We didn't go through what the paper looked like. So here's the second of those two papers where they looked at the mechanism. And here we go. This is the first of those two papers on Neuropillin. Okay, so we've got 5 to go. Let's do it. So this next one I think is a wonderful picture story. So they took little swabs and put them on the armpits of people who had COVID. So these are COVID patients, and so they were getting a sample of what it smells like, just the total, I don't know, metabolic bouquet, of a COVID-infected patient. They're putting them in little tiny cylinder filters. How nice. Let's see, and These are who's a good boy. That dog is, and that dog is as well. They tried it out with different dogs. So they had drug-sniffing dogs, explosive-sniffing dogs, I think, and they're trying to train up COVID-sniffing dogs. And I think these are their COVID-sniffing dogs. You'd have to dig into it. Here's the cone of olfaction. Yeah. Here's the little glass jar where the sample is going to be placed. And so this is how they're trying to standardize a dog smell test to see whether the dogs can recognize it. It turned out that the dogs that they trained up to recognize this were able to recognize COVID patients, and far more often than by random chance. And so that's the one down there. And so here's all our different dogs, and some of them were a little better than others, maybe. Yeah, Oslo was pretty good. We saw a picture of him or her up there, yeah. I don't know. So there you go. I didn't think it was possible, but there it is. Yeah, COVID-sniffing dogs. What do you know? Strange world. All right, next one. We've got in this one, so this is a report that the zinc finger antiviral protein, ZAP, yeah, is when you knock it back with a siRNA, so an interfering RNA, it actually allows the virus to grow more. So ZAP is restricting virus growth to some extent, which means that here where we have lots of ZAP, the virus growth is going to be lower. Here where we have less ZAP, the virus growth is going to be higher. And this is played out over and over again across this half of the figure. And so it's not an extreme restriction, but it is something that is slowing down the virus. And this is something that slows down various other viruses as well. This is the paper on that one, if you need it, is this one right here. So there you go. Okay, next one is a paper looking at individual sequences of coronavirus from inside of patients. So they're looking at the quasi-species, like this cloud of mutant viruses that ends up cooperating, even though there's no overall, organization to the cloud. And they're looking into it and saying, okay, where are there deletions? So we know there are changes up and down, and a lot of these changes are random, but we're seeing some interesting deletions. And so some of these deletions are right here at the S1, S2 cleavage. So this is the cleaved end that would be recognized by Neuropillin right here, and this is the cleavage by furin. Here is, let's see, the cleavage site. The fusion peptide is actually right over here, so they've drawn that wrong, and that may be something that gets corrected. But they've got a deletion right in there, so it's either deleting part of the fusion peptide if they made the FP right, or it's deleting part of the cleavage site if they made the cleavage site thing right, and who knows. These deletions are not the number of amino acids that are being deleted. This is like they're numbering their deletions. They range from very small to very large, 2 to 18, I think, nucleotides, they were saying, are deleted in each one of these things. And so most of these deletions are going to be inactivating, but what they're showing here is similar to what is seen in persistent mouse hepatitis virus. When it goes into, I think in particular, neurons in the spinal cord, mouse hepatitis virus has been shown to actually delete big chunks of its own spike, which allows the virus to basically persist there indefinitely. And if it had the spike on, it would keep spreading, and you would get immune response, and it would be able to spread like that. This is a different aspect of the virus when it goes in and buries itself and essentially lives with that mouse for the rest of its life. We do not have any evidence that the SARS Coronavirus 2 can do this in people, but these are the sorts of mutations that you might expect to see if SARS Coronavirus was doing this in people. So this is an interesting little lead-in to whatever comes next. I'm curious to read those studies, and that would be, very, very interesting, I think. Okay, and so if we need to see the author and title. There we go. Boom. All right, next one. So this is looking at the new Ferret model. I think this is the second paper, possibly the third, to publish a Ferret model. But it's a nice sort of overview of the Ferret model. And so you give ferrets the virus. When they give them, 5 times 10 to the second, so 500 infectious units, they only got 1 out of 6 ferrets infected. So the regular infectious dose, mean infectious dose, is higher than that. When they gave them 5 times 10 to the 4th infectious units per ferret, they got all of them infected. They did not try different concentrations in between. And so our real infectious dose for this virus in ferrets is somewhere between 5 times 10 to the 4th and 5 times 10 to the 2nd. And whatever, somebody else will find the exact range. But this is useful information. Doesn't mean it's exactly the same in humans, but it gives us a ballpark in which this virus is going to work. And so the ferrets get the virus. They control the virus after a while, and then the ferrets get better. So it's not a lethal challenge model, but it is one where they get sick and they will get some lung damage over time, and they'll have a temperature increase. So there's their poor little temperatures going up. And here's some of the lung damage. It's, I don't know, it's noticeable, but it's not extreme from what I've seen of the human samples. They also tried in this paper to reinfect the ferrets. And I think in the last one of these multi-paper reviews, we looked at a paper with cat coronavirus where they tried to reinfect cats that had been infected a month ago and were unable to infect them. Well, in this case, in the ferret model, they were definitely able to infect the ferrets. You can see this is the viral RNA, but the ferrets did not develop severe lung disease or even as severe as the original one. So the ferret model seems to be a fairly mild one. They get a mild form of the disease, but potentially useful in some ways. It replicates some, but not all, of the clinical characteristics of actual COVID-19. So yeah, there you go. If you're thinking about ferret models, that's useful. And last paper we're going to cover today, they are looking at what are the minimum set of proteins that you need to make virus-like particles. And they came up with the S, the E, and the M protein. And you could actually probably have gotten particles with just the E and the M protein. That's the case with regular SARS coronavirus, but they're really inefficient. The efficiency gets boosted a bunch when you add that nucleoprotein. That's just like, I don't know if it was a 10-fold or 100-fold increase in efficiency of budding. And they put the spikes on the outside, which you don't actually have to have there, but you do have to have them if you're going to try to deliver these into another cell or use them as a vaccine. And I think they're using this as a potential vaccine strategy. So instead of putting the spikes on the outside of a different kind of virus like VSV, they're saying, why not just put the spikes on the outside of a virus-like particle from a coronavirus that you can grow up in great batches, and then they have one that they are tapping over here. So this is atomic force microscopy. They're tapping on it, and it's about this tall at first, and as they keep tapping harder and harder, eventually they pop it, and here's the inside sort of gushing out over there. And they've flattened the poor little virus. I just thought that was amusing, and they put that in there. So there you go. That's the paper. That's our wrap-up. 13 papers in however many minutes this was. Thank you very much, and I hope this was of some use to somebody.